Hi, everybody, and welcome to this week's lecture. What we're going to be looking at is going to be sex determination and sex-linked inheritance. Now, by the end of this week, we should be able to understand both of those. When it comes to this week's lecture, it's going to be broken up into three separate parts. The first part, or this lecture, is going to be all about that sex determination. In our second lecture, we're going to be talking about sex-linked inheritance in humans. And then in part three, or that third part, we're going to be looking at a lot of examples when it comes to sex-linked inheritance and exactly how to do those Punnett square problems, because there are going to be some very specific rules we need to follow in order to track exactly what traits are linked to what sex chromosome and exactly how those are going to transfer out and how we're going to get our genome phenotypes based upon those Punnett squares. But let's go ahead and continue and check out our sex determination, exactly how we can dictate whether we're going to have XX, XY, male or female. So when it comes to determining sex, one of the major things we need to understand is exactly how is that going to happen? What do we need in order to get the two sexes? One of the very first things we need to remember is it's all based upon our sex chromosomes. So if you can think back to the previous lectures where we have X and Y, possibilities, depending on the combination, is going to give us whether a male or a female. So typically, chromosomal sex is determined at fertilization when a sex chromosome from either sperm, which is going to be either X or a Y, joins with the sex chromosome from an egg, which is always going to be an X. This combination, if it's an XX, is going to give us a female. And then if we have an X and a Y, that is going to give us a male. Now, a couple important things to remember and understand is chromosomal sex does not always match up with our gonads or phenotypic sex. Now, what does that exactly mean? When it comes to gonadal sex, that refers to the gonads, testicles, or ovaries present in an individual. And then phenotypic sex refers to the structures of the external and internal genitalia. So again, although typically when we have XX, they'll have ovaries, and then typically when we have XY, that's going to give us our testes. That isn't always the case. There are some exceptions to the rules where the chromosomal sex will not match up with our gonads or gonadal glands or our phenotypic sex. But typically, when we have an XX and XY, we end up with either testicles or we do end up with our ovaries. Now, let's see exactly how we can get our X's and Y's, where they're coming from, and exactly how we're getting those to come together to determine the sex of the offspring. So again, in humans, our chromosomal sex is going to be determined at fertilization. Now, one thing to remember is sperm can either have or carry an X and a Y. Now, when it comes to eggs from females, it's only going to be an X. Now, if you go and look at this graphic over here, you can see these are going to be the possible male sperms. You can see we have XY. Remember, through meiosis, we go from diploid to haploid. So that's why we end up with these four possibilities. Looking at this over here, you have about a 50% chance of contributing an X, and then you have a 50% chance of contributing a Y. And again, if we end up with an X and a Y, we're going to end up with a male. And if we have XX, that's going to be a female. Now, one thing to remember is for our eggs, it's always going to be 100% X. And that's going to be a very important factor because the X chromosome is an absolute must. We absolutely have to have an X chromosome because it's going to contain much more genetic material, and we can't actually survive with YY. So you can see that we have 50% XX, 50% YY for our possible male sperm right here. And then you can see we have 100% for female, which is always going to be XX. So again, if we have XX come together, we're going to have a female. And then if we end up with an XY, that is going to be a male. So let's go ahead and test our knowledge to see exactly how we can kind of do this and how we can predict what our offsprings are going to be. So I'm going to go ahead and give you about 5 seconds to pause this video, give this a try, and then we'll come back together and check it out. All right, welcome back, and let's go ahead and check out this problem. So it says, how is chromosomal sex determined? You can use Punnett squares to examine the inheritance of X and Y chromosomes and the SRY gene, which we'll talk about in just a second. So when it comes to sex chromosomes of a mother, remember that's going to be a female, so it has to be XX. For father, it's going to be XY. So if we go ahead and place these over here and here, in this case, the mother will be our P1, and then our father will be our P2. So we're going to go ahead and put XX over here for a female gametes, and then over here we have our X and Y. We go ahead and do our Punnett square. So again, remember these drop down here and here. So I'll put X here. Again, this is going to drop down here and here as well. So X. This is going to go over here for both of those. So X. And this is going to go all the way down over here. Y. So looking at this, we have two of these, which are going to be XX. And then we have two of these, which is going to be XY. So in this case, what is a percent of a child born with will be chromosomally a male? Remember, in order to get a male, it's XY. We see 2, so we have about a 50% chance. What percentage of children born will be chromosomally female? In this case, XX. So we have about a 50% chance of it being a female. So you can see it's a perfect 50-50 split for our Punnett square, again, giving us a 50% chance of a male and 50% chance of a female. And that's what that should look like. All right, so in that previous problem, it asked or mentioned something called the SRY. So the SRY is going to be something that's very important. Remember, when it comes to sex chromosomes and when it comes to determining the sex, we always must have an X. You can see over here that our X chromosome is here and here. You can see that this is drastically bigger, about 50% larger than the actual Y chromosome itself. Again, the reason why is because there contains lots of genetic material in here that we need to have in order to survive. So we always know when it comes to sex chromosomes, there's always going to be at least an X chromosome. Now, there are some certain genetic disorders where it's going to be, let's say, XXX, maybe XYY. Again, you might have extra ones or missing ones for the second, but you will always have an X chromosome when it comes to sex. So let's go ahead and continue and talk about the SRY gene. So when it comes to SRY, this is essentially going to be the protein that's going to dictate a male. You'll notice over here that it is only going to be found on the Y chromosome. You can see for the female, we don't have that over here. And essentially what happens is that SRY gene is going to go to our SRY mRNA, which is going to help create that SRY protein. In that presence of that protein, that's what's going to cause a male to develop. Okay, so again, the SRY protein is a transcription factor. So that protein regulates transcription and gene expression. SRY activates genes and promotes testis formation, marking the first steps toward male rather than female. So again, that is going to be another differentiating factor, is that it's going to promote the transformation or formation of a different gonad versus a female. Because there is no SRY gene, it's going to go ahead and produce ovaries rather than testes. So how does that work? How does gonads or the glands develop in humans? So following fertilization, an embryo begins to develop, and at first, its sex organs are a genderless gonad. Okay, so that's going to be very important, and that's going to be known as a bipotential gonad. Basically, what that means is it's kind of like the foundational portions or foundational parts to making either the testes or the ovaries. So basically, it's going to be a small, thick ridge of tissue near what will become the abdominal. And at this stage of development, the gonad is, again, a bipotential gonad because the tissue is capable of developing in either the male testes or female ovaries. So this is what that's going to kind of look like. You can see we have that bipotential gonad right here. And you can see that, again, it looks very, very similar to what the testes are going to look like or what the ovaries look like. Again, think about this kind of like a foundational piece. It just needs that specific signaler, the SRY gene, to either understand whether it's going to become, again, testes with that present of the SRY. And if it's absent, then it's going to go and develop into ovaries. Okay. So how is phenotypic sex determined in humans? Now, when it comes to this, in XY embryos, testes produce a hormone known as testosterone. This testosterone is going to stimulate development of male internal and external surfaces. So you can see over here, once we have that SRY, again, if we go back to this, we have that XX. And again, remember that Y is going to carry the SRY gene. That SRY gene is going to go ahead and produce that protein. And in that presence of protein, we're going to start developing testes. So once we start developing that, we're going to have testosterone present. And with that testosterone present, what's going to happen is male phenotypic is going to develop. You can see we have the testicles here, vas deferens, some vesicles, prostate, urethra, penis, scrotum, all of those things. Again, this is going to be that external portion, and this is going to be that internal portion. Versus the ovaries, you can see that without the SRY gene and without that protein, that testosterone is not going to be produced right here. And because of that, female phenotypic development is going to happen. So now you can see that again, the ovaries were developing because of the absence of the SRY gene. And then you can see that we now are going to have a clitoris, a labia, and then a vagina. So this will be our external, and then this will be our internal. So again, in XX embryos, ovaries are not going to produce testosterone. And because of that absence in testosterone and the presence of estrogen, it's going to stimulate the development of female internal and external sex structures. So this is kind of a flow chart. Definitely remember this because this makes things a little bit easier to understand exactly what's happening. You can see we have an egg with an X chromosome. Remember, in humans, we always have to have at least one X chromosome. And then if the sperm is going to contribute a Y chromosome, again, what's going to happen is that Y chromosome is going to have the SRY gene. The SRY gene is going to produce that protein. That protein, again, we have our XY, and then that protein is going to stimulate the development of testes. That testes are going to start producing testosterone, and that testosterone is going to give us our male sexual structures. Okay, so again, this helps break it down. You can see this is our chromosomal sex. In this case, we have our XY. We can see we have our gonadal sex, which is going to be the testes due to the SRY stimulate. And then our phenotypic, what we can see in this case, male sexual structures. In this case, it would be the penis and the testicles. And then if the sperm is contributing an X chromosome right here, you can see we now have the absence of that SRY. So we have our XX chromosomes, and because we're missing this SRY, they're going to start to develop ovaries. Ovaries do not produce testosterone, so there is no testosterone. Instead, they're going to be producing estrogen. So because of that absence in testosterone, this is what's going to cause that development of female sexual structures. All right, and that is going to conclude the lecture for today. So as always, if you do have any questions, comments, concerns, please feel free to reach out, and I look forward to seeing everybody in part 2.